So, hello everyone. Good day. We're still on the social uses of genetic discovery, and we're on topic 5, which is the paternity testing using the blood types and DNA. So, before we start, let us look on the meaning of paternity testing. Well, on the other groups, they have already discussed this paternity testing using the fingerprint. Now we are going to look on paternity testing using the blood types and DNA. So, occasionally, situations arise in which people require concrete scientific evidences of parentage, whether it be their own or that of someone else. In most instances, maternity is easy to determine. Before surrogate motherhood, so when we say surrogated, a woman who becomes pregnant by artificial insemination or by implantation of a fertilized egg created by in vitro fertilization for the purpose of carrying the fetus to term for another person or persons. So before this, a surrogate mother, yung hanap ka ng iba para siya yung mag-bear ng anak mo. Sila yung magbubuntis para sa anak ng iba. Bago yun naging posible, the woman who gave birth to a child was obviously the child's gestational, genetic, and legal mother. And this continues to be true in vast majority of cases today. Unfortunately, questions of paternity aren't so easy to answer. In order to make the determination of fatherhood, scientists almost always work backwards from child to the potential parent to ascertain the actual nature of the relationship. In the past, so before, they use they base it on the specific phenotypes. When you say phenotypes, we are talking about the character, the outward character. It's like, for example, if he is tall, like that, the height, the body build up, and but in paternity testing using these specific phenotypes, it is the specific blood types of the child, and they are using this information to either rule in or rule out possible fathers. However, this system presented a number of problems, not the least of which was that it often yielded inconclusive results. Thus, since the 1990s, the more common approach has been to consider the presence of particular genotypic markers. So we are going to discuss further about that genotypic markers as we go on to our presentation. And the presence of particular genotypic markers when attempting to establish fatherhood, and in a handful of cases, motherhood. So before, it's the specific phenotypes, and since 1990s, they are now looking and they are using the information about these particular genotypic markers. So next, we are going to see the use of blood types in paternity tests. So, as what I have said a while ago, DNA fingerprinting was developed by Alec Jeffries in 1984, and it first became available for paternity testing in 1988. Before this sort of DNA analysis was available, blood types were the most common factor considered in human paternity testing. Blood groups are a popular example of Mendelian genetics at work. After all, there are numerous human blood groups with multiple alleles, and these alleles exhibit a range of dominance patterns. Today, the best-known blood typing system is ABO typing, which involves presence of antigens on red blood cells that are encoded by the ABO locus on human chromosome number 9. So, ito yung ginagamit ng paternity test, the ABO blood type. Kasi maraming mga types ng blood. We can consider the Duffy, the KID blood group system. So, in the ABO system, the A allele and the B allele are codominant, and the O allele is recessive. We've already discussed that on the previous topics. Thus, if a person's ABO blood type is O, he or she has two O alleles. If, however, a person's blood type is A, he or she has either two alleles or one allele and one O allele. Similarly, if a person has type B blood, this indicates the presence of either two B alleles or one B allele and one O allele. Finally, some people have type AB blood, which means they inherited both A allele and a B allele. In cases of questioned paternity, ABO blood typing can be used to exclude a man from being a child's father. For example, a man who has type AB blood could not be a father of a child with type O blood because he would pass on either the A or the B allele to all of his offspring. So, there are also limitations of using ABO blood typing because despite their usefulness in this regard, ABO blood groups cannot be used to confirm whether a man is indeed a child's father. So, let's look on this example that because of this and several other factors, it took the legal system some time to trust blood typing. For example, in a famous case in 1943, the starlet Joan Barry accused actor Charlie Chaplin of fathering her child. Although blood tests definitely showed and tell that Charlie Chaplin is not the father, they gave meron pa rin silang binigay na considerations kay Joan Barry at sinabi nilang kailangang suportahan pa rin ni Chaplin yung anak nila, si Carol Ann. So, back then, there was only one method of determining the paternity of a child, a blood test. So, back then, meron lang blood test lang ang pwede nilang magamit para ma-prove na si Chaplin ay father or not nung anak ni Barry. So, blood was drawn from Chaplin, Barry, and baby Carol Ann. 3 doctors weighed in on the results, with which one coming to same conclusion. Pare-para silang conclusion. Chaplin was not the child's father, which prompted Barry's own lawyer to state that 3 distinguished medical men, preeminent in their fields, have decided that Mr. Chaplin is eliminated. We must and do abide to their conclusions. So, when it comes to the medical conclusions, disregarded na sa pagiging father si Chaplin. But then, very naging persistent si Barry na bigyan pa rin ng parusa si Chaplin. So, dun nagkaroon ng parang change pagdating sa paternity sa paternity laws dun sa Amerika. Kasi merong nangyari na parang voting, merong jury na nag-vote kung bibigyan ba ng responsibility si Chaplin sa anak ni Barry or not. So, over time, the use of additional blood, so hindi in-honor yung result ng ABO blood groups. Kaya there are issues pa din when it comes to ABO, when it comes to using the ABO blood groups in legal system. So over time, the use of additional blood antigens such as those associated with the MN. So MN is the blood group system that is under control of an autosomal locus found on chromosome 4. And RH systems, this system is for classifying blood groups according to the presence of the or absence of the RH antigen, often called the RH factor, on the cell membranes of the red blood cells. So these rhesus and MN blood groups refined the use of blood typing for both paternity and forensics. However, such blood groups were only about 40% effective in ruling out a man as a child's father. And fortunately, in 1970s, testing for human leukocyte antigens, or the HLA, added a distinguishing feature that made it possible to rule out men as fathers with 80% effectiveness. So the genes responsible for the HLA system are involved in antigen presentation to T cells. The HLA system is highly polymorphic with more than 3,200 different alleles identified so far. This is according to Robinson 2003 and Williams 2001. Although this vast number of alleles causes headaches for cell and organ transplants, the multiplicity of genotypes the HLA system provides in tens of millions makes it ideal for consideration in identity and paternity testing. So, the utility of paternity testing. Interestingly, improvements in paternity testing over the past several decades have not only led to an increase in the accuracy of test results, but also to expanded application of various testing methods. For example, the DNA technology, such as DNA sequencing, polymerase chain reaction, DNA cloning, and gel electrophoresis, has gotten more precise. It has become possible to determine paternity using DNA from grandparents, cousins, or even saliva left on discarded coffee cup. Such DNA testing is clearly an important part of criminal investigations, including forensic analysis, but it is also useful in civil courts when the paternity of a child is in question. The widespread availability of paternity tests on the web and in the neighborhood drugstores is also indicative of the civil demand for this technology. However, it is important to note that such direct-to-consumer or DTC tests will not stand up in court because there is no way to prove whose samples were analyzed. So that's their limitation. Hence, DTC testing is most often used to assist in making a decision to initiate litigation or to simply provide peace of mind in matters of questionable paternity. In broader applications, advances in paternity testing mean that people who are adopted now have more direct means to confirm their biological identity or to find their birth parents. In addition, parentage testing is often an essential tool in proving immigration status in cases of family reunification. And so we're going to summarize it all, that for years, questions of paternity presented a significant challenge to scientists and potential parents alike. During the first half of 20th century, researchers often turned to people's ABO phenotypes when such issues arise. However, ABO blood group information could only be used to exclude potential fathers rather than confirm the presence of parental relationship. So take note of that. Consideration of additional blood markers such as Rh antigen, Mn antigens, and HLA greatly increased the effectiveness of paternity testing over the next few decades, but it still left significant room for error. So it's not 100%. Thus, with the dawn of DNA analysis and sequencing techniques in the 1980s and 1990s, scientists increasingly began to look at people's genomes when questions of fatherhood arose. This approach proved exceedingly useful. In fact, current markers-based methods on analysis yield test results that are both 99.9% accurate and applicable in a variety of settings. With the ongoing advancement of DNA sequencing and analytical technologies, we will no doubt continue to see an increase in the utility of these tests, as well as in the availability of detailed genetic services to the general public. So I'm hoping that you learned today. So thank you.